Assalamualaikum, my name is Fahana. Today, my group and I will be presenting our assignment on the three recent advances of vaccine. So, in the past few years, the emergence and re-emergence of disease has greatly challenged the efficacy of the existing vaccines against specific disease. The traditional way to produce vaccines are based on live attenuated organisms, virus vector, and subunit vaccines pose a risk of regaining their pathogenicity under certain immunocompromised conditions. Therefore, a lot of effort has been made by the scientists to develop new vaccines. The use of several latest vaccines, which will be presented by me and my group members, are said to improve vaccine efficacy. So let's go to the first one. So to increase the immunogenicity and stability of subunit vaccines, several approaches have been used, such as virus-like particle VLP vaccines and nanoparticles and vaccines. VLP, which stands for virus-like particles, have a similar structure to virus. They are composed of all or part of the proteins that can be arranged in several layers and forms a viral capsid. However, they lack any viral genetic material. Due to that, they are unable to replicate and causes infection in the recipient, which enhance safety during manufacture and administration procedures. So as illustrated in figure 1, VLPs also have repetitive surface patterns which stimulates the immune system to produce high cellular and humoral immune response. Figure 2 shows schematic of VLP vaccine production. In brief, one of the methodology to produce VLP vaccines may include, firstly, the nucleic acid of gene interest will be engineered into plasmid expression vectors encoding an antigenic protein. Next is the expression of viral proteins using a variety of expression systems such as bacterial, yeast, insect, and mammalian cells. After that, the viral structural proteins self-assemble to produce VLP and display the antigenic determinants of target pathogens on their surface. Next is the nanoparticle vaccines, which are produced by chemically cross-linking protein antigens and carrier with the help of linker molecule to increase immunogenicity and decrease degradation of the antigens. These carriers can be lipid-based, polymeric, or metal-based molecules. Studies have shown that nanoparticles have been used for vaccine delivery with several advantages, which includes enhanced protection against premature degradation, good stability, and improved adjuvant qualities. Nanoparticles provide protection for the antigen's native structure by encapsulating antigens within nanoparticles during transportation to target cells. Nanoparticles also improve antigen delivery to antigen-presenting cells and enhance duration of antigen presentation to immune cells by decorating nanoparticle surface with antigens.
Okay, so next I will move to the next part, which is DNA vaccines. So DNA vaccines contain DNA that codes for specific proteins or called as antigens from a pathogen. Then this DNA is injected into the body and will taken up by cells. So there are various methods of DNA vaccines, but these two are the most popular DNA vaccines, which are gene gun and also electroporation. For gene gun, the DNA plasmid is coated with gold particle or tungsten microparticles, and it will taken up by cells via a blast of pressurized helium gas from a gene gun. The result of gene gun is it will increase humoral responses but limited in allowable dosage. So the second one is injection in saline, or it can be assisted by electroporation. The plasmid is taken up by permeabilized skin followed by an electric current. So the result of electroporation is that it will maximize DNA vaccine delivery efficiency.
Next, I will continue with RNA vaccine. The development of mRNA started much later compared to DNA due to the inherent stability of mRNA, and a few methods have been introduced to improve its stability. The first method is RNA engineering. The method involves the addition of 5' cap and cosec sequence and 3' poly A sequence to mimic the structure of endogenous mRNA to avoid detection by innate immune sensors and adding modified nucleosides to increase RNA stability. The second method is thermostable RNA. It involves the freeze-drying RNA and incubation with various biomolecules to increase the RNA stability even at high temperature. The third method is dendritic cell vaccine. For this method, RNA is transfected into dendritic cells in vitro, which can then activate CD4 T cells and stimulates antibody production in vivo. Last but not least is self-replicating RNA. The structural proteins in the genome of the RNA virus are replaced with the coding sequence for the antigen, and the viral polymerase can keep producing RNA, which amplifies and maintains the amount of the extinct protein. That's all from me.
Okay, next I will proceed with the vaccine for immunosuppressed individuals. Okay, first, what is immunosuppressed individual? Okay, immunosuppressed individuals is a suppression of the body's immune system and its ability to fight infection and other diseases. It can be result from certain diseases such as AIDS or lymphoma or from anti-cancer drug. Okay, the example of immunosuppressed individuals are young children, the elderly, and immunocompromised for medical reason. Furthermore, the cause of immunosuppression in each population are vary, and their consideration for underlying mechanism should be taken into account. Okay, the example of factor that cause the variation in immune system are age, sex, and also a specific environment factors. Okay, for young children such as infants and neonates are considered to be immune suppressed due to age-specific immune system development. This is due to decreased expression of T helper 17 supporting cytokines by TLR receptor and also increased expression of anti-inflammatory cytokine. Okay, the function of T helper 17 cell to defend against extracellular bacteria and fungi as well as the development of autoimmune disease. On the other hand, the immunosuppressive phenomenon observed in the elderly has been referred as immunosenescence. Immunosenescence known as the decline of immune system with age, which lead to a higher incidence of infection, neoplasia, and autoimmune diseases. Okay, immunosenescence can cause defect in innate and adaptive immune system, degradation of lymphoid architecture, dendritic cell have reduced uptake of antigen, and decrease the number of naive T cell and beta cell. So that's all for my part. I will pass to the next presenter.
Okay, now I will explain about the use of adjuvant in the vaccination process among the immunosuppressed individual. Firstly, we need to know what is adjuvant. Adjuvant is a substance that enhances the immune response to the presence of antigen. So basically, the adjuvant can only be used in a subunit vaccine as they do not contain any potential viral genomic element that can increase probability of developing disease. Some of the adjuvants that commonly used are aluminum salt base or alum and oil in water base, and each adjuvant has different advantage and disadvantage. For instance, the aluminum based adjuvant can trigger strong humoral immune response, primarily mediated by secreting antibodies specific to antigen, while it generating weak cell mediated immune response. And there are also other new adjuvant that scientists discovered, which is LPS derivative based ASO4, which being used in hepatitis B and HPV vaccine and found to increase the cell-mediated immune response in patients with end-stage renal disease. So basically, the adjuvant is one of the greatest achievements in medical history because it is not only meant for immunosuppressed individuals, but the main purpose of the adjuvant itself to increase and strengthen the immune response after the injection of the vaccine. Thus, we can improve the use of vaccine by reducing the number of antigen and the number of the immunization. So this simple choice will favor the choice to improve the functionality of future vaccine against numerous diseases. As a conclusion, these new technologies in the vaccine development has also expanded the idea of what can be done with vaccination by expanding its reach to the area that have never been addressed before, such as cancer treatment and also the neutralizing of drug abuse.
So this challenge as well as opportunities will ensure that the vaccine development will remain in science to combat the new upcoming emerging disease. I think that's all from our group. Thank you.